Before I introduce our keynote speaker, first of all, let me welcome you all to this sort of 7th edition of what we now know as the joint RECOMB Regulatory and System Genomics with DIM Challenge Symposium or conference. It's been an amazing experience to see this meeting grow over the last 7 years with Gustavo and Manolis, and to see some of the real impact that, for instance, some of the work in the DREAM challenges has had. As you're sure, the people who attended yesterday's portion of the meeting have. But let me just remind you that when we started in Boston, we had a couple hundred people, and now we've grown to about 400 when we do this conference in the States. A little bit less because, unfortunately, some of our postdoc and students that are on a sort of student visa cannot travel abroad. But I think still we reach about 300 people registered for the conference this year in Toronto. And so welcome to Toronto, 6th annual version of the joint meeting. And actually, even before it was a joint meeting, there were separate meetings that were 2 different RECOMB sponsor sort of satellite conferences. So as usual, I think we bring together an extraordinary group of scientists. We're going to have 16 really highly respected keynote speakers. I think just for the fun of hearing what they have to say, this makes this conference really a seminal place to be. About 253 DREAM Challenge submission, I think this year more than any time before, 81 oral communication and 97 posters. So just a very brief set of words on DREAM. I think DREAM has been with us for the last 3 years, and I think it has become sort of the thing that keeps us honest about the work that we do. As you know, this is a conference track that was really inspired to the CASP meeting that in structural biology has set some absolute standards to be able to measure how well we do computationally in predicting protein folding when you actually already have the data from the crystallographic structures. And we are trying to accomplish the same thing in systems biology and regulatory genomics, and obviously this is a much more complicated issue because I think there's nothing more precise than the atomic level measurement of structural elucidation, while, for instance, when we talk about interactions in a complex biological systems, establishing what would be the ground truth becomes a little bit more of a complicated matter. And so I think it's been extraordinary that even within those limitations, Gustavo and now more recently Sage have been able to really bring such an amazing group of people together. And just want to see the number of sort of submission and participants has grown very steadily. And now a lot of people even are excited about the fact that they can showcase their experiments and their results when normally they would not have been able to have so much visibility. But now that there's a completely objective metric for determining how well they're doing, they're coming to the surface much more effectively. This is the last year, as you can see, basically almost double the number of submissions to DREAM. And DREAM has also been very strongly supported by the NCI. I think as we move forward, one of the things that we're trying to do at the NCI is to make sure that the enormous wealth of data that comes out of clinical studies can now be used, for instance, for challenges such as the ones that the DREAM program has put together. So as Diane said before, again, thanks to the sponsors that have made this possible. This is a great, one of the great challenges of putting together a conference of this size is to find companies and scientific organization and sometimes public government organization that can help you put it together and sponsor it. And it's been an absolute pleasure to work with many of these organizations in organizing the conference both this year and in previous years. So we have a really, having a little trouble seeing both the screen or here. So we have a really exciting session for today and tomorrow. We're going to start with Peter Jackson, and I'll say a few words to introduce him, but we're going to have then Peter Sorger in the afternoon today, And then we have, as you know, we lost an absolutely phenomenal scientist and a great friend, Tony Paulson, and we thought that he had already agreed, in fact, at the time, to give one of the keynotes talks here at the conference. And so we're going to have a memorial lecture that will be given by Jeff Rana, and then finally, to close the day today, we're going to have Bernard Palsson giving us a talk on metabolism. And then tomorrow, the meeting will reopen in systems biology for the second day with Olivier Pourquet. Olivier is an absolutely terrific developmental biologist, and I thought that having somebody that presents that perspective, by the way, he does developmental biology in a way that is extraordinarily related to sort of systems type approaches. I thought that having that kind of biological perspective may also be of great interest and sort of motivate a lot of the people in the audience to maybe think about working in that area. And then finally, we're going to close with Brenda Andrews, Toronto representative, who has been really probably one of the people that have started the field with Tim Hughes in terms of the seminal work she's done in East. One thing that I want to say is that it's been remarkable how 6 years ago when we did this to start, I was trying to put together sessions, and among those sessions, we were trying to figure out whether somebody was interested in thinking about drugs or druggability of cells using sort of systems approaches. And there probably was one presentation in that area, and that is it. We were thinking about representing work for disease and development using systems biology. And again, maybe one or two of the posters or the talks had to do with that particular topic. And I think you can see the success of this field and how it's evolved just simply by looking at how much work has become really impactful and change the way we understand biology, not change just the way in which we think about solving biological problems. So, and I think this is reflected. I was just on the web going through some of the centers that are represented in systems biology. And you can see the list of now department in system biology all over the world has become really substantial. I don't know why it's not showing, but anyway. So with this said, switch back. Maybe this will work. Okay, so basically this said, I would like to introduce the next speaker, actually the first speaker and the opening speaker for our systems biology track, Peter Jackson. We tend to be very sort of low-key in introducing speakers at these conferences because we tend to concentrate on the science. But I have to say that Peter has been a phenomenal researcher because he's been able to combine both an absolute rigor in the basic science component and actually made things matter in terms of even thinking about developing drug and driving some of the major drug program at Genentech. Now he's back at Stanford. He's concentrating on some of his very elegant work on cilia. And when I first heard of cilia, I thought, wow, that's a narrow field. And when I actually figured out what Peter was doing in signal transduction in cilia, I said, wow, this is affecting every single disease, every single phenotype that we can think of, and that has a cellular sort of framework and background. So I don't even know how he finds the time to shoot the trilogy of The Ring or any of the Hobbit movies, but however he does it, I would like to just welcome him to the stage.